Alright, so this is my biweekly update for June 10th, 2022. So obviously my entire focus for the past week has been revising the COVID manuscript. I think I've gotten through all of the main difficult points of revision for the most part, and then what remains is incorporating some citations here and there, going through the document one or several additional times and rewording certain sections to make it clearer, and um, other minor suggestions, such as incorporating citations to support the targeting of certain NSPs that are not in their bioactive conformation alone, like NSP 7 and 8. So most notably, my focus was the protein-ligand interaction discussion and a revamped version of the Swiss-ADME drug likeness section, which I will show in a little bit. But I was experiencing some problems initially, particularly in regard to Pymol. As I mentioned in a message on Friday, it was behaving strangely. It selected residues, I believe, which were too many residues, selecting nonpolar residues, and also showing hydrogen bonds or polar contacts to the carbonyl oxygen, as I showed in the message on Friday. So I wasn't really confident about the results that Pymol gave me, and I started to look around. I found BioVia Discovery Studio, but it required Windows, so I couldn't use it. So then you suggested Chimera, and I conducted all the analysis in Chimera again for all of the 18 promising leads. I looked at each one of them, the best docked pose for each one of those, and I determined the linkages they had around hydrogen bonds. And all of the hydrogen bonding interactions predicted by Chimera were also predicted by Pymol. However, Pymol predicted many more in addition to the ones predicted by Chimera. And Chimera also didn't give me the problem with two carbonyl groups showing a polar contact between each other. So that was reassuring, and I feel more confident in Chimera. And those are the results I put in the manuscript. So, yeah. However, I still found that I could make figures with Pymol. It's easier. Pymol seems to be more user-friendly. And, well, not on the next slide. I put them in the manuscript, but I will discuss this further. Strangely enough, Pymol never showed any pi cation pi interactions of any sort for any of the 18 compounds, though this might not actually be a mistake on its part. It just might be that they simply don't exist. I didn't see an option for showing pi interactions in Chimera, though I did look for it. So these are the final results for Chimera predictions and Pymol predictions. Chimera predicted fewer hydrogen bonding interactions for every single protein ligand pair. All these on the left column are the 18 promising leads that were revealed to have one or fewer violations in Swiss-ADME. So yeah, Chimera seems to be more strict about the requirements it uses to detect hydrogen bonds. One notable exception, there are a few differences. For instance, for RAIN, Pymol doesn't predict lysine as an interacting residue, but Chimera does. But for the most part, all of the residues predicted by Chimera are also predicted by Pymol. Just Pymol seems to be more lenient. The interesting standouts, the 6XM4 with columbamine, which didn't have any hydrogen bonding interactions, also didn't have any hydrogen bonding interactions in the case of Pymol, and neither did 6XIP, or rather 6YHU with jetrorzine. So that seemed to be consistent. What I'm really focusing on the interaction analysis or general trends, I'm mentioning the fact that for 6YHU with its different ligands, the most common residues that showed up are valine 66 and glycine 64. They seem to form a key part of that binding site due to their hydrogen bonding interactions. And I'm also going to focus on RAIN and 6 or 7 NIO because RAIN had one Payne's violation indicating potential promiscuity, and it also has 9 hydrogen bond locations, which um, may potentially inform a conclusion that it could be a promiscuous molecule. Um, but yeah, really, all I can say is general trends like valine and glycine are common for certain protein structures, and certain I really didn't see a trend between increasing number of hydrogen bonds and increasing Rosetta energy unit binding affinity. Obviously, there's much more than just hydrogen bonding that goes into that, but I did state that all binding interactions below -18 REU had at least 2 hydrogen bonding interactions, so they did play a role in stabilizing the compounds. Again, yeah, Pymol seems to be more lenient regarding the criteria by which it samples the H bonds, so that's why I focused on Chimera, and I used Pymol for figure making. Because I mentioned the binding energies in the analysis of the interaction section, I was thinking of actually adding it to the table. I actually made a table with the binding affinity data. I pull it up here. I have this table needs to be revised a little, but yeah, the docking energy score, the hydrogen bonding residues, and all of the compounds. I was also thinking of adding the length of the hydrogen bonds. Chimera automatically gave me the length between the residues and the ligand for the hydrogen bonding interactions, so I thought that might be informative. I think I'll add it just for completeness. I might not actually make any analysis with it, or I can. Okay, so this was another major revision, replacing Figure 1 with something more comprehensible. Figure 1 initially, according to Reviewer 4, was not very comprehensible, at least in regard to actually learning what the workflow entailed. So I replaced Figure 1 as shown. Perhaps I should get the actual PowerPoint slide out. That might be a good idea. This is the new Figure 1, and as suggested, I put numbers designating where, at what stage each particular stage happens, and then Roman numerals to correspond to certain points in the text. And I upgraded this description as well to make it significantly more clear. Hopefully this is better than what previously was. I think it is. It made much more sense after reading it numerous times. And I also upgraded the description of the whole workflow. There was one in right at the end of the introduction, right here, right immediately before Figure 1. And I also upgraded this one to use more clear language. Yeah. Just for reference, if I can get it, the old Figure 1 was this. And now the new one is this. If I can find the old description was not very good. This was the old description, obviously in much less detail, and no numerals corresponding to different points in the diagram. And then the new one. Okay, so also Reviewer 4 requested that we include a table with all of the protein structures used, their resolutions, their common names, and the references to the papers that they were sourced in the RCSB. I did exactly that. The references still have to be put in, but I have the DOIs for all of them, and Ray is going to meet Tuesday, and we will put all of the citations in for good measure. And update the bibliography with all the new citations and everything, and it should be done. But yeah, this is, I specified starting coordinates as opposed to active site location. There might have been a misunderstanding with the reviewer because they said put the location of the active site, but we don't have the active site coordinates, so, and I mentioned in a little subscript at the bottom of the table. I'll actually go to it directly. Yeah, so there's that point, which I made. And I say they are the centers of 3D regions sampled by the ligands during docking. I made sure to clarify that, and I'll clarify that in the response to their comment as well, noting that it's not actually the active site we're necessarily sampling. And just an additional point, obviously 6XM4, we didn't sample the full structure. We used the RBD from it because no unbound structure of the RBD exists or existed at the time of the analysis. And still I couldn't find any now, so just made that additional point there. But yeah, that's an additional table. What else? Okay, so this is a big thing. Reviewer 4 made a bunch of comments regarding the score function analysis section. They were confused. They asked us to explain the statistical analysis, and most tellingly, they asked us to determine whether or not there were covalent bonds between the protein ligand complexes, and five of, and I wrote this was the response I'm writing for that. So 5 of the 10 main protease structures did have covalent ligages with their ligands. And I actually looked in PyMol to see if it was the case. At first glance it wasn't, but when I actually zoomed in and used show sticks by reds all within 5 of ligand, they showed up, so it was pretty concerning. So 5 out of 20, I mean, we didn't actually specifically look to see to what extent that changes the analysis, but I really don't feel comfortable, and I told this to Ray and he agrees with me, we really don't feel comfortable including this analysis anymore. It really doesn't fortify any claim we try to make in the paper. And ultimately we changed this with a section simply explaining our score function selection process based on the results of the Myler lab paper. So I will go to there right now. I changed all of that and simply mentioned the results of the Smith et al. Paper, Myler paper, which demonstrated that Rosetta ligand pre-Talaris 2013 outperforms the others in several tests and is the best for small molecule screenings. Because ultimately that's what we tried to prove with the score function analysis. Reached the same conclusion, but our analysis was faulty. So in the methods, the change isn't much, but in the results and discussion, we are thinking of deleting the entire Rosetta score function validation section because of this analysis. And this also, some of the wording in this section, I understand could be confusing to some, and we couldn't really figure out a way to reword it well. And also, referee 4 asked a ton of questions about this, not just the covalent linkages. He asked about like statistical analyses and justifying and whatnot. So this isn't to be lazy, but I think it would be easier if we were just to get rid of it altogether, and it wouldn't hurt us in doing so. So yeah, that was the major point of contention that I kind of struggled over, but ultimately, I don't know. I think it's the best thing, but please let me know if there's anything else we should consider. Yes. So, okay, after these, the score function analysis taken care of, interaction analysis mostly taken care of, the writing's there, just the figure needs to be tweaked a little. Drug likeness section. Oh yeah, drug likeness section. Reviewer 1 mentioned there's no explanation of the results, so I beefed up that section quite a bit. Made many changes. Before I just mentioned that some leads were poorly soluble, and then I mentioned some of the implications of poor solubility. I mean, I thought, I may have thought they were obvious, but perhaps they just wanted better explanation. Yes, it impacts biodistribution. I added mention of PGP substrates, how that also impacts biodistribution, rewording, and then more in-depth analysis between the comparison I made in the supplement between these three in table S8 and doravirine and Paxlovid. So yeah, just beefing up this section, actually explaining what things like pore solubility and PGP inhibition cause, rather than just simply stating that those things happen as indicated by Swiss ADME. So I think that's what they were going for. Hopefully that clears it up. Whatever. MMGBSA and MD. That's not. I was thinking about it for a while. There's web servers that sort of do it, but not what I want. There's one that does MMGBSA for protein-protein complexes that I found. I haven't found one for protein ligand complexes. And either way, I'm assuming they're not very robust. They're pretty old servers from like 2010, so. MD simulations. I mean, really what I want that can show is just an RMSD versus time plot that shows the ligand is stable in its location over like, I don't know, like 4 nanoseconds or something. That's really all I can get for it. But I don't think it really will add anything of value, and it likely will be too much time before I can actually get the results and write everything up as needed. So yeah, I'm most likely going to forego this. Almost certainly. Okay, so questions. Yeah, this is the only thing I could think of now, but since currently I'm not directly involved, but I will be once I finish this thing next week, early next week, fingers crossed. Most of the work on the antibody methods chapter. What can I write specifically for it? Maybe some of the background on the introduction regarding Q beta. I don't know if the actual like sequence assembly is important, 10x genomics and all that. Or maybe I'm not the best person to actually write about it since since Sam has been working with it for over a year now. So yeah, since affinity maturation isn't on there, I'm just wondering what specifically can I write for it, or maybe I'll just be relegated to doing like figures and editing work, which I'm fine with, but I don't know, maybe there's like a specific topic I can research and actually contribute to like a complete section on it. So yeah, that's about it. I'm glad the thing is nearing its completion, and hopefully we'll have another pair of eyes looking at it once all the revisions are in place, and on Tuesday the whole group will look and see if the revisions matched the suggestions. All right.